and welcome to our coffee chat on patient-centric sampling. I am Naamah Maundrell, Senior Editor of Bioanalysis Zone, and I'm delighted to be joined today by three experts who all have a real passion for patient-centric sampling. So firstly, could you introduce yourselves?
Hello, I'm Stephanie Cape, Scientific Director from Covance Laboratories. Very excited to be here today to talk about patient-centric sampling.
Hi, and I'm Shane Needham, co-founder and Chief Scientific Officer of Alturas Analytics. Welcome to New Orleans.
Melanie Anderson at Merck. I'm a principal scientist there and very excited to talk about patient-centric sampling today.
So to kick off this coffee chat, let's start with the basics. What is patient-centric sampling and why is it so important?
So, patient-centric sampling is sort of the idea that we can adjust the way we sample so that it's more convenient and easy for patients. And so sort of that's the driver here, is that we want to make things better and easier for patients. I think there's also certain indications where it enables us to get better data, more data, richer data sets, so that has a lot of value add in healthcare.
Yeah, when I think about patient-centric sampling, I think about our culture in general of becoming a convenience culture, frankly. There are no movie stores by my house anymore. I don't, travel to a movie store and rent a movie to bring home and watch. I want to have that available to me immediately, where I want it and when I want it. And especially if I'm in a situation where my health is compromised, I definitely want that flexibility for myself and my family to be able to have those samples taken at home where I'm most comfortable. Mm-hmm. So to me, it's really all about the patient and what's best for the patient. And it's our obligation as scientists to enable that.
Yeah, I agree with Stephanie and Melanie fully. And I think the future of healthcare is really going to be patient-driven. And it's becoming a topic more and more. But as scientists, we are supposed to develop methods and techniques and give tools to make sure that patients can be the drivers of healthcare instead of somebody that may not know as much about them. And convenience is a huge one as well. And so I think it's just part of the process of patients really taking over. The more and more information that we have now on the internet and the web and so on and so forth, is that taking over their own healthcare and being responsible for it. So that's part of it.
I also think there will be some tangential advantages in terms of the ability to recruit patients for clinical trials faster, the ability to retain patients, the ability to reach new patient populations in remote geographical areas, as well as geriatric populations, pediatric populations, the ability for us to get data that we've never been able to get before.
One of the things that I think about too when I visit different areas, whether it be a hospital or so on and so forth in my travels, is that I see oncology patients for a lot, and I'll see them, no hair, wrapped up, so on and so forth, and they're very sick, and they're getting out of a taxi cab, for example, or an Uber, and they are walking into the facility to take a blood sample. And that just seems ludicrous.
Yeah.
Why not be able to take that blood sample at home? That might be a 24-hour sample or a 48-hour sample. Who knows what it is? But why not be able to take that at home? And so I see it's not only convenience, but it's about the patient's health, and their lifestyle and their quality of life. And that's huge for the future. And that's really what's going to drive, I think, the future of healthcare.
Well, and I think too, so if we can improve access as well. So currently, like our clinical trial paradigm is very site-centric, right? So people have to be geographically co-located wherever that trial is happening. And if we can sort of change this so that people in rural areas and other places can have access to oncology trials through more patient-centric sampling, that also is a huge win.
Sure.
So after discussing why patient-centric sampling is so important, can we now look at how patient-centric sampling has evolved and how we got to this point in time?
So, I think so if you go back far enough, the very beginning of sort of DBS, dried blood spot sort of technologies, right, where people were driving for smaller sample volumes, it's really newborn screening is the major win there, right? So from the 1950s on, we were doing this, and we do this routinely in every hospital in all America, right? And this is a huge win to detect metabolic disease in young children, in neonates and children in the hospital even before they leave the hospital. So it's a sort of proven approach that we can do microsampling, we can do dried sampling. It's made a huge shift in healthcare. And I would say that, maybe 15, 20 years ago, DBS and dried blood spots were sort of pushing into therapeutic monitoring, and then also, pharma and CROs were looking at this, can we do this for PK? Can we do this for PD? And so a lot of the groundwork has, I think, been laid for, can we detect this analyte? Can we detect it in a state that someone could actually collect it at home? So I would say that much of the work has been done, right? So we know we can do this. We know what tests we need to do. And so sort of how do we now take this technology, which can be pretty disruptive, and push it into a space that's going to make, really make our approach more patient-centric, right? For all those reasons that we said before.
Yeah, I agree completely. And I mean, you talked a little bit about DBS, and we all know that DBS suffers in the PK space from hematocrit issues. Automation can be somewhat challenging, but some of the new scaffolds and some of the new devices and techniques are overcoming those analytical challenges with defined sample volumes and enabling us to take this space forward. So I think that there's a lot happening in the industry right now in the device manufacturing space as well that is really pushing this forward and allowing us to think about implementing on a broader scale and on this widespread clinical scale.
Yeah, I agree again. And I think that the challenge is not necessarily in the bioanalytical lab. The bioanalytical lab has always delivered. We've always done things and improved techniques and created new tools and methods and so on and so forth. So we have to ask ourselves the question, why is this not being implemented, for example, maybe on a wide PK scale? And those probably aren't questions that we can always answer. But because it's not the technology that's limiting. It could even be just the skepticism involved in, well, it's different than plasma, it's different than whole blood, it's different than serum. So we might have to think differently. And so I don't think in the bioanalytical lab, we can make the tools, we can make the methods, we can make the techniques. It's why is it not being implemented? Anytime we have to force somebody to use our techniques and our methods that they're not really wanting to implement, that's an issue. We have to decide, well, who wants this and needs it, and where's it going to fit? Because there's never ever going to be one tool that works for everything, and that's okay. So where are the areas that, for example, dried blood spot or patient-centric sampling will fit? Neonatal, oncology, sparse PK, limited areas or geographical areas. So I think we have to be careful to make sure that we're not trying to say as analytical chemists that this is going to work for everything, but how can we create the tools to make it as easy as possible for everybody when they decide to implement it for their studies?
Well, and I also think the conversation has been driven by the bioanalytical community, and I think that needs to shift. We want our clinical monitors and our physicians and our project teams and people in healthcare coming to us and saying, this is the value add for patient-centric sampling. Can you do this for us, right? And so in many ways, I think we're just at the point where we're trying to shift who's driving this and who knows that this is going on and what we can do there. I think one of the downsides that this was driven by PK is that PK is so hemmed in by, sort of our regulatory expectations, as it should be. But, would there be a space that would be a little more amenable to this for just PD over time, or, like therapeutic monitoring, some of these other approaches, I think would be a little more flexible for pushing it in the beginning, and then PK could follow.
I think it's interesting that you mentioned that it's being driven by the bioanalyst and that we need to get the clinical teams on board. Just reflecting on the idea of dried blood spots and the idea of dried blood spots in the preclinical space, and that's something that bioanalytical teams are very, very comfortable with. We have quite a bit of experience with, and so as you mentioned, I think as bioanalysts we understand that we know what we need to think about, right? We know that we have to ensure stability, that we have to ensure recovery, that humidity may be an issue with dry times. All of the pieces of the puzzle and the experiments that we need to put together to ensure data quality, we understand all of that very well due to our experience. But a lot of that came from the preclinical space. So it may be in that realm, in that regulated GCLP, a little bit newer to our clinical teams to think about implementing something so different.
Well, and I think another hurdle as we move, it's one thing to take a sample in a clinic, regardless of whether it's plasma or dried blood or whatever. And then if we're actually talking about people taking samples at home, we really have to work through the details around the sample quality, right? So how is that sample taken? Was it done properly? And so that's where the device manufacturers come in. And it's, really important that we have, ways to do this that are really simple and really easy.
And they have to be controlled ways, so we can monitor that scientifically. And we all know science, for example, but the normal patient doesn't, nor they shouldn't have to. And so they don't realize that you have to document everything exactly when you take the sample, and the quality of your sample, and when you took it, and how much volume there is. That all matters. And we've made some tools around that, and there's other technology that we can do to try to control that, but it's still difficult when we're used to the science. We create these methods and tools and techniques all the time. The normal patient does not, nor should they have to. It should be simple to them. However, implementing that in an at-home setting is not just straightforward. There are a lot of things we have to think about. So where are the niches that we can think about where it really, really is a need for the tools that we've developed?
Yeah, I agree. And in terms of implementing the right tool for the right situation, I also, really feel that there will be more technology solutions that address some of the things that you just mentioned in terms of when the sample is taken. And if you are going to punch a button to take a blood sample, can that button also trigger the timestamp? The devices to be barcoded, dropped in the mail, shipped back into the labs, things like that, I think are well in the works.
So we've touched upon how patient-centric sampling has evolved, but where are the current opportunities that you're seeing?
Well, so I think in pharma, obviously we've mentioned specific disease states, right? So oncology, migraine is another that's a big one where people don't take the medicine all the time. They only take the medicine when they have a migraine. They're not going to be in the clinic. Things with long half-lives. So there are specific disease states where there's a huge win for at-home sampling. And then I think a big driver in pharma for this as well is that we have, these well-controlled phase 1 trials. Phase 2 are a little less well controlled, and then we go into phase 3 with very little PK data, right? So we're making decisions on, safety and efficacy without even knowing if people actually took that drug. So, are they having viral rebound because the drug doesn't work, or are they having viral rebound because, they forgot their medicine that day? And so understanding what's happening in that space is really important for project teams to really understand how our drugs are working, right? So that's a big driver there as well. So I would say, disease state, and then secondarily, our POP PK large trials. How do we actually get a better feel for what's going on in that space?
Yeah, I think it's been proven that we can do this with neonatal therapeutic drug monitoring and small studies, and even preclinical, and even for large population PK. It can be done. Why isn't it being implemented on large PK studies? Why? And because that's really what drives a lot of the bioanalysis, our large clinical PK studies. So what is the hesitation there? And does it need to be there? Do we need to have patient-centric sampling? Certainly for certain disease states, let's think about anxiety, for example, where somebody has anxiety, they take a medication, it may or may not work well for them. It's kind of a one-size-fits-all solution, in a lot of ways. It's not patient-centric, truly. Healthcare, I think, is changing, that we're trying to make it more patient-centric, and this is one of the ways we can, is that then they could take a blood sample after they take that medication to see exactly what dose they should get. But right now, you don't have that. You have a multi-person trial, thousands of people, and you take time points over time, and you're trying to look for a one-size-fits-all solution, which is not patient-centric at all. So I think that that's where this will help with certain disease states.
And I think as we start to realize the benefits of that and see the gains in data quality and what we can learn about different disease states and dosing regimens, that's where the industry will really take off and gain momentum.
So I think it's clear that there are some real niches where patient-centric sampling is or will be having like a real impact. What are some of the other advantages of patient-centric sampling and why we need it?
So within the clinical trial space, I like the idea of the dried sampling. So there are patient-centric sampling devices that offer wet sampling that will collect a microsample and it stays in the blood form or is processed to a plasma form. But with a dried sample, you can eliminate cold chain, which has some major benefits. When you're talking about remote geographic locations where there may not be centrifuges or -70 freezers, being able to collect those samples and transport them back to the laboratory at ambient temperatures can have some huge advantages.
I mean, I think of just, running a bioanalytical laboratory is that one of the things with a dried sample that has been alluded to already is that we don't need a centrifuge necessarily. We don't. Think about the freezers. We don't need freezers. That takes up so much space in our laboratories. It takes up energy. It influences the environment, so on and so forth. It's a huge cost. So, the more and more we can implement dried samples in the appropriate form, I think it is better for the overall science, and it will also. It's possible that the return on investment is better because it's a lower cost, because you don't have the centrifuges, you don't have the freezers, so on and so forth, and that's what we're expected as scientists to deliver, is lower cost to the patients, and that's really what it's going to be about. The current system is not sustainable. It's too expensive for anybody to handle, and so hopefully we can change that paradigm.
So I think from what you're saying, there's a real need for patient-centric sampling, and as technologies develop and become more advanced, more will become available. But you've kind of alluded to it already, but what are the hurdles that remain for actual implementation of it?
So, and I come at this from like a pharma perspective. So sort of, I think when we interface with our teams, we are these large organizations with certain workflows, right? And changing those, it gets tricky. It's hard, right? And that's why I said, it is disruptive. So, sort of even figuring out all the details about how you train the clinical site, how you train the patient, now take that times 100 in all different languages all over the world with a late phase trial, right? So these are like major hurdles for implementation that seem pretty easy. And then when you actually go to do this, communicating that you need desiccant to a clinical site, across the world in an effective way, and making sure 6 months later when the trial's still ongoing that they're still aware that this is important, those are major hurdles. So just the execution piece, I think, has been our major issue. The science works. We kind of know the workflow that we need there, but even how the data is going to flow back into our systems as far as like a timestamp, how is that going to work? And those are things that we're trying to work through, but it's these sort of cross-functional, lots of different people. It's not just the bioanalytical space. So I think there needs to be a big return on investment if you're going to be putting all of this energy into kind of changing your workflows. But I do think that the benefit is there. It's just a matter of thinking a year or two ahead and making these changes.
Yeah, I agree, and if I think about the challenges in just analytical chemistry in general, historically, when you're making an analytical measurement for any type of a solution, is that's usually not where the challenges are, because it's a very distinct, easy to define problem, and it's usually solvable with the right personnel and the right mindset and the right ideas and resources. But it's implementation. So you have, let's just think, clinicians and phlebotomists that have been clinically trained generationally for the last 50 years, 60 years. That's probably how long it's been. It hasn't been that long where we've really had phlebotomists, right? Now we're going to train them to take dried blood samples in a clinic. That's huge. That's a huge change. And we've heard about those challenges in pharma. when you try to implement this on a worldwide scale. So it's not going to happen in 1 or 2 years. And maybe the new clinicians have to be trained maybe in a different setting or in a different way instead of the typical, IV. I don't know. I don't have the answers, but the challenges aren't in the bioanalytical lab. The challenges are, why is it not being implemented on a worldwide scale? And because there are challenges with people not accepting it. And that's okay. There must be a reason. So we have to figure out why.
So I think that there are challenges to change all through the spectrum, right? So definitely challenges, at the point of sample collection. There are some regulatory, not challenges, because, in general, the regulators are very, very supportive of these new technologies, but there's a little bit of additional work in that bridging studies need to be conducted if you're using microsampling for PK and comparing that back to historical plasma data, for example, as well as the bioanalysts are definitely up to the challenge, I agree, but there are some additional considerations when you're thinking about microsampling and sensitivity limitations, making sure that you can reach the sensitivity levels that you need, as well as that your compound is stable in that dried matrix ambient condition, as well as recoverable from whatever device. So I completely agree. The bioanalysts are up for that, but it's something that requires inertia and upfront investment in every portion of the program. But I agree with Melanie that once you overcome that return on investment is just enormous.
So I think you all agree that implementing the technology is expensive and disruptive. In what areas do you think patient-centric sampling will be worth the investment? I think you've kind of touched upon neonatals, oncology. Any others?
Oncology, neonatal, therapeutic diseases that are immediate. For example, anxiety, migraines. What are some other ones that you can take? You can take an immediate sample, so then you can get basically feedback from that person on what the exact dose should be. Because right now we just say, this is the typical dose for a typical person, and this is what you're going to take, and because it works for most people. Well, that's not the right way to make patient-centric healthcare. And so any of those types of diseases that have an immediate effect and I guess a short half-life, so to speak, where they're not, a migraine may last a day, but it may only last 2 or 3 hours. Take samples at that time, so we could do patient-centric sampling, at-home collection, send your sample in, and at least be able to review that data and say, this is what we think is going on.
Well, and I would say too, we kind of are all, we're drug level people, so we think in terms of like drug level, right? But I think there's this huge shift in the industry to understand, broad, protein levels, metabolomics. There's all of this like push to better understand what's actually occurring in the human body over time. And I think we're just at the tip of the iceberg in what we can get out of that stuff, right? So that may be an area that's really going to push and revolutionize the patient-centric sampling. People are going to begin to expect that the industry is pushing to better understand what's actually occurring in our body, right? Not just how much drug is there and did viral load drop, but all of these other pieces of what's going on in our body at any given moment. And patient-centric sampling there could potentially be a huge win.
No, monitoring those biomarkers. And you're seeing this. You're starting to see this in professional sports, for example. It's big in the UK. Where they take these athletes and they're monitoring them all the time, but you can't necessarily draw their blood because you're not with them all day. So they take at-home samples and they send them in. So I think that's a very, very good point. And patients could be the same thing. A regular patient can be the same way. So you can know their whole biomarker, get a lot of that biomarker data from one sample.
And in fact, you're starting to see more companies that are doing this, where you can take a finger stick sample, do a dried blood spot, send it in, and get a read on your metabolic state, or what are your lipid levels, things like this. So some of this is going to be driven from sort of the wealthy well and what they're trying to get out of the analytes that they can detect in the dry
state. It might be the wealthy well, but not necessarily. You can already do some of these things, whether it be hormones, lipid profiles.
And they're pretty affordable.
You can go down to Walgreens. It's cheaper than going to the doctor. I do it. You go down to Walgreens, and it's a dry blood spot, and you get your lipid profile, so you get your cholesterol, your VLDL, your LDL, your HDLs, and all that. And in a week, less than a week, without ever going to the doctor.
Yeah.
And so the future is happening,
starting.
you know, and there's some bioanalytical lab there that's, you know, actually analyzing the sample. So it's happening, but it's, you know, it's very distinct. So.
And I also think if we're really pushing for patient-centric sampling and sort of, and there's lots of different ways this is happening in the industry where they're trying to pull out of sort of, people have to go to this specific site, right? Sort of the siteless clinical trial. We have to think drug level, we have to think biomarker, and you know, we're beginning to see some clinical chemistry, like the cholesterol and in these other spaces pushes there. So if we could take one sample, right, and get a read on your PK and your PD and your safety levels, that's huge. And I think that's where it's going. It's just a matter of time. But in the meantime, there's a lot that's going on that needs to adjust.
And it might be more than one drop of blood.
Yeah, true.
But I think, you know, all of this is exciting because, like you mentioned, the clinical chemistry is going there and the at-home wellness checks. And so it's a lot of people coming at this from a lot of different perspectives, all recognizing the huge potential and collaborating and working together. And so, you know, what can we learn from each other in that space? So, yeah.
I mean, I think a little bit of a concern is the Theranos story. So, you know, I mean, I think obviously this woman wasn't a scientist. She wasn't interested in actually getting reliable data out of these things. But I think what it did show is the appetite for this type of approach. And, you know, we as scientists and people in the healthcare industry, this does have such potential to give us better data and make better decisions and things like this.
Well, and I think we just discussed more cost effective. If we, if any of us as a patient can just walk down to the drugstore and pick up a kit and not have to make a doctor's appointment, not have to go in and read our levels, and especially with some of those pretty simple and I guess simple tests like HDL, LDL, cholesterol, whatever, there's enough information to know we know what a healthy state and non-healthy state is. Okay, and so we're not trying to define anything revolutionary here. We're just trying to see if we're in a certain range, and so it's happening, and it's more cost effective, and it actually lets the resources that are intensive in clinics and hospitals, so on and so forth, deal with things that they really should be dealing with. Because right now the system's not sustainable. There's too many people and not enough places to go, and it's too expensive. And we all know that. That's a question. So there's many different answers, but none of it's sustainable. So we can be part of the solution to try to make this more cost effective for people.
So if we move the conversation on now to think practically, how do we analyse these samples to make sure we get good quality data from them?
I mean, so what we have seen, and I think you guys have probably seen this too, we see a lot of issues with, you know, stability over time, and how do we track what these samples have been exposed to? Even if they are taken in a clinic, how do you know what shipping, what went on? So that's a big one. Another is extractability over time. So we've seen in certain sorbents that, you know, recovery will change and shift and things like this. And so those are questions that you want to think through almost ahead of time. And so what we have implemented, because we've started to see this in some of our trials, is almost like you know, there's this testing you do for phase 1 and maybe the beginning of phase 2. And then there's like the thinking that needs to go into a long phase 3 trial, and what do we really need to understand in that space with respect to stability. So I think from a bioanalytical perspective, that's what sort of our limits are. I think the hematocrit thing has pretty much been solved. So.
Yeah, those issues have been solved, and I think what we tend to forget is that those are the same issues we have with plasma and serum, for example, or whole blood, or whatever matrix that we're actually analyzing. Is we have stability issues, extractability issues, so on and so forth. And one of the ones that we do have that we don't have as much in general with in typically a larger sample size, liquid-wise, is that we can have a limit of detection or limit of quantitation problem, because you can only punch out so much of that dried blood spot. And so that's why, you know, a lot of vendors or a lot of people always ask, well, have we reached the levels of sensitivity that we ever need, and that we don't need any better lower limits of quantitation? And the answer for analytical chemistry is always no. No. When we get to single molecule detection for all methods, then we've reached what we can reach. But that's the goal, right? That is the goal. I mean, because always better instrumentation, always better limits of detection are always going to make things easier for the bioanalytical lab. And just because one molecule, you can get a detection limit of whatever, 10 nanograms per mil, 1 nanogram per mil, 1 picogram per mil, doesn't mean you can get that with another one. And so there are limits with the size of the actual punch that will limit your lower limit of quantitation.
Yeah, and you had mentioned that we've always had these issues with plasma or serum in traditional sampling mechanisms and techniques as well. I just think we're really good at managing those at this point. We're very comfortable managing those. It's not unusual for us to have an analyte that maybe isn't very stable in plasma through the sample collection and handling process, and we know what additives to try and how to stop enzymatic breakdown. And so as we think about how this field will mature, and we think about having more tools in the toolkits in terms of, are there additives that can be added to the dried blood spot sampling to ensure stability as well as improve extractability? I think that will definitely help us apply the technology to a wider variety and range of molecule classes.
And I think that manufacturers are starting to respond to these needs, and you're seeing different sorbents coming out. I think a thing that's unique to out of clinic sample collection is what was that sample actually exposed to, even in shipping. Better understanding that and doing the appropriate testing there is really important too, and that's regardless of analyte, right? If stuff is falling apart and you're making decisions about that, that's a bad decision. So.
Yeah, and that's a place where we really need, as bioanalysts, to have that appropriate experimental design.
Yeah.
When we think about shipping at ambient temperatures, what does that really mean?
What is ambient?
Yeah.
Mm-hmm.
Right, because even just being in a plane, that's a lot of exposure to potential freezing.
Right.
Yep.
Yeah, it doesn't even have to be winter. So those kinds of things are things we should be thinking about and doing ahead of time. And you don't want to be in the middle of your trial and find these things out, especially if you're pushing new technology and making organizations shift their approaches.
Yeah.
That's a bad place to be.
So if we look towards the future now, what advances are we already seeing, and what is the time frame of implementation for some of the technologies that we've talked about today?
So I think we're already seeing huge advances in the clinical diagnostic space, in the clinical chemistry space, in terms of people being able to take their own samples at home, ship those back into the laboratory, perform wellness checks, making those sorts of laboratory diagnostics more accessible to the population. Lots of movement as well in the device industry. Everybody is collaborating and behind this.
Yeah, I think we already talked about some of those, but whether it be at our local pharmacy where we can see some of these clinical measurements that are routine, cholesterol and hormones and so on and so forth. So we're seeing some of these things implemented. As far as timeline goes, I think that's an interesting question, because if you look at any technology that's been developed, we look back and say, oh my goodness, that was revolutionary. So let's just take the car, for example. Let's just say that it was about 1900 where it really started to be, becoming, I guess, okay to drive a car, and the technology was there. There were still people in horse and buggies in the 1920s and 30s. But we look back, 130 years later, and we say, oh my goodness, that was revolutionary, and it was so quick. I bet you they didn't think it was quick back then. And I said, this technology is going to be the same. It's going to be just little by little, step by step, incremental changes until we get to a point where we're going to, maybe 50 years later, we're going to look back and think, oh wow, those were some big changes, over a 50-year period. But over a 1-year period, 2-year period, 3-year period, doesn't look like there's much movement, but there always is.
I think digital health is here, right? And we're seeing this more and more. I think pharma is starting to push for this. We're beginning to see that on a wider scale. I think healthcare in general is doing a lot of this already in different pockets for different applications. So it is here. I think it will be curious to see sort of how it grows and matures in our space.
Yeah, definitely.
As the technology continues to develop and improve, what do you think we can actually expect to see in the next 5 to 10 years, and how is patient-centric sampling impacting patients' lives now?
So we're already seeing trials with patients collecting samples at home, like we're seeing that across the industry. So that is sort of here. Figuring out how to like marry the technology in the best possible ways so that we're engaging with patients at the appropriate places and all of that. You know, that's still, we're all working on that still. But no, I think it should be an exciting time. I think this will be the bubble 5 to 10 years where we see that we really can get better data out. We really can better understand, you know, drug levels and people's biomarkers, you know, landscape, and better make decisions for their health. So I think it is here. It'll be, you know, any change is disruptive and it takes time. I think the bioanalytical labs have already done this. Like, we've already made the changes in the workflows and the spaces. I mean, we can do this. But I think, like we said, the hurdles sometimes are in the bureaucracy behind the scenes, but we're making headway in that space too. So people want this. I think and it adds so much value to healthcare.
Yeah,
it's here.
The biological lab will change as needed, as it always has. I think they'll be incremental. We've already know how to extract these. I mean, but Stephanie's mentioned maybe how to stabilize them better or make better extractability.
Sure.
Little tweaks like that, but automation-wise, and I think we've got pretty much that figured out. But I think what I'm excited about is that I think this is a good opportunity for the patient to be the consumer and the consumer to be the patient, and they're going to drive where this goes. And that's important for sustainable healthcare, because they're going to say, well, it was too hard to collect, or it was, oh, there were too many time points. What else would they say?
Too painful.
Too painful.
I forgot.
Yeah. And so then we're, as scientists,
which we all do.
Yes. And then as scientists, we're supposed to develop methods and techniques to make sure that we deliver to them. And that's what a consumer-driven society does. And that's what's going to get us really, really good healthcare, is when the patient becomes a consumer and the consumer is the patient.
And what I hope is that, you know, as we migrate away maybe from the site-centric approach, the sort of economic limitations that people have to being able to be in a trial will also be alleviated and hopefully just gone altogether, right? So that people can't, you know, don't have barriers to enrollment just because they can't get to that site or they can't afford to, you know, take off work to go back and give samples every week, those kinds of things, because those people are out there and we're not really meeting their needs with our current clinical paradigm, you know, trial paradigm. So that's something that I think needs shifted.
So I think kind of looking towards the future is a great point to close this coffee chat. Thank you all for your contributions today. I think we've highlighted some really impressive devices and technologies that are already having an impact on patient lives, and hopefully we can continue to collaborate to see more in the future. Great.